This is an example of a somatic cell. Because there are two complete sets of chromosomes, this cell is a diploid. Most of the time, a cell is an interphase. Interphase includes 3 stages. During G1, the cell begins to take in materials and grow. In the S phase, the DNA is replicated and checked for any errors. Finally, in G2, the preparation for mitosis begins. Once the DNA is copied and the cell has enough energy and materials, the M phase is ready to move forward. M phase includes 2 distinct parts, mitosis and cytokinesis. The first stage of mitosis is prophase. The chromosome begins to coil and condense until they become sister chromatids combined by a centromere. This is commonly referred to as a chromosome. Matched pairs in a diploid cell with different alleles are referred to as homologous chromosomes. The nuclear membrane begins to break down and spindle fibers begin to become visible from the centrioles. Finally, the centrioles begin to move to opposite sides of the cell. At the end of prophase, the spindle fibers are attached to sister chromatids. This is what pulls the sister chromatids to the middle of the cell. The next stage of mitosis is metaphase. During this stage, the chromosomes are lined up in the middle of the cell, called the metaphase plate. The chromosomes are then attached to the spindle fibers from each side of the cell. During anaphase, the centromeres are split apart and the chromosomes begin to separate and are pulled to opposite sides of the cell by spindle fibers. The cells also begin to become elongated. The final phase of mitosis is telophase. The chromosomes are now opposite sides of the cell. A nuclear membrane starts to form around each new set. The spindles continue to get smaller and break down. They are not 2 different cells yet. During cytokinesis in animal cells, a cleavage furrow forms within the cell and pinches off the plasma membrane to complete the separation. In plant cells, a cell plate forms and acts like the cell wall in each new cell until the cell wall is able to fully form. The cell cycle is a loop. Once 2 new cells form, they are then able to start interphase and eventually divide again.